Because insects are cold-blooded animals, their rate of development is more or less dependent on ambient temperature. 48 bodies exposed to large amounts of sunlight will heat up, giving the insects a warmer area to develop, reducing their development time. An experiment conducted by Bernard Greenberg and John Charles Koenig with the use of rabbit carcasses to study accumulation of degree days found that with temperature ranging in the mid-70s to high 80s the amount of developmental time for maggots was significantly reduced. 49. In contrast, bodies found in shaded areas will be cooler, and insects will require longer growth periods. In addition, if temperatures reach extreme levels of cold, insects instinctively know to prolong their development time in order to hatch into a more accepting and viable climate in order to increase the chance of survival and reproduction. Furthermore, insect activity and colonization patterns can also be influenced by the length and intensity of solar exposure. Because the increasing temperature speeds up their development, insects are more likely to be active and colonize a body more quickly in places exposed to direct and extended sunlight. In comparison to shaded locations, this may result in a quicker succession of insect life and disintegration stages. On the other hand, because of the lower temperatures, shaded areas might have slower rates of insect activity and decomposition, which would delay the processes of insect colonization and decomposition. 50. Air exposure. Air exposure can have a significant impact on insects and the determination of postmortem interval (PMI). Hanged bodies can be expected to show their own quantity and variety of flies. Also, the amount of time flies will stay on a hanged body will vary in comparison to one found on the ground. A hanged body is more exposed to air and thus will dry out faster, leaving less food source for the maggots. The presence and behavior of insects on hanging bodies can vary. As the body begins to decompose, a compilation of fluids will leak to the ground. This area is where most of the expected fauna can be found. Also, it is more likely that roe beetles and other non-flying insects will be found here instead of directly on the body. Fly maggots, initially deposited on the body, may also be found below. 46. Geography. According to Jean-Pierre Megnin's book Le Faune des Cadavres, there are 8 distinct faunal successions attracted to a corpse. While most beetles and flies of forensic importance can be found worldwide, a portion of them are limited to a specific range of habitats. It is forensically important to know the geographical distribution of these insects in order to determine information such as post-mortem interval or whether a body has been moved from its original place of death. Caliphoridae is arguably the most important family concerning forensic entomology given that they are the first to arrive on the corpse. The family can be found worldwide. Chrysomia rufifecis, the hairy maggot blowfly, is a forensically important member of the family Caliphoridae and is widespread, however it is not prevalent in the Southern California, Arizona, New Mexico, Louisiana, Florida, or Illinois regions. 51. Flesh flies fall under the family Saprophagidae and generally arrive at a corpse following Caliphoridae. Unlike Caliphoridae, however, members of this family are able to fly in heavy rain. This key advantage enables them to occasionally reach a body before Caliphoridae, affecting the maggot mass that will be discovered. Flesh flies are globally distributed including habitats in the United States, Europe, Asia, and the Middle East. 52. Beetles are representative of the order Coleoptera, which accounts for the largest of the insect orders. Beetles are very adaptive and can be found in almost all environments with the exception of Antarctica and high mountainous regions. The most diverse beetle fauna can be found in the tropics. In addition, beetles are less submissive to temperatures. Thus, if a carcass has been found in cold temperatures, the beetle will be prevalent over Caliphoridae. Weather. Various weather conditions in a given amount of time cause certain pests to invade human households. This is because the insects are in search of food, water, and shelter. Damp weather causes reproduction and growth enhancement in many insect types, especially when coupled with warm temperatures. Most pests concerned at this time are ants, spiders, crickets, cockroaches, ladybugs, yellowjackets, hornets, mice, and rats. When conditions are dry, the deprivation of moisture outside drives many pests inside searching for water. While the rainy weather increases the numbers of insects, this dry weather causes pest invasions to increase. The pests most commonly known during dry conditions are scorpions, ants, pillbugs, millipedes, crickets, and spiders. Extreme drought does kill many populations of insects, but also drives surviving insects to invade more often. Cold temperatures outside will cause invasions beginning in the late summer months and early fall. Box elder bugs, cluster flies, ladybugs, and silverfish are noticed some of the most common insects to seek the warm indoors. 53. In general, insects are poikilothermic animals, thus meaning their level of activity is substantially dependent upon their surrounding environmental conditions. An increase in the temperature will result in an accelerated metabolism of the insect, hence resulting in an increased activity. 54. Modern techniques. Many new techniques have been developed 55 and are used in order to more accurately gather evidence, or reevaluate old information. The use of these newly developed techniques and evaluations have become relevant in litigation and appeals. Forensic entomology not only uses arthropod biology, but it pulls from other sciences, introducing fields like chemistry and genetics, exploiting their inherent synergy through the use of DNA in forensic entomology. In order to improve the precision and dependability of insect-based evidence analysis, forensic entomologists nowadays use a variety of cutting-edge technologies, such as stable isotope analysis and DNA analysis. These methods have broadened the field of forensic entomology by making it possible to identify insect species more precisely, pinpoint their geographic origins, and draw important conclusions about the circumstances surrounding a death. Some of the most important contemporary forensic entomology techniques are examined in this section along with how they are applied to criminal investigations. Scanning electron microscopy. Fly larvae and fly eggs are used to aid in the determination of a PMI. In order for the data to be useful, the larvae and eggs must be identified down to a species level to get an accurate estimate for the PMI. There are many techniques currently being developed to differentiate between the various species of forensically important insects. A study in 2007 demonstrates a technique that can use scanning electron microscopy, SEM, to identify key morphological features of eggs and maggots. 56. Some of the morphological differences that can help identify the different species are the presence, absence of anastomosis, the presence, absence of anterior and posterior spiracles. 57. The cephalopharyngeal skeleton as well as the shape and length of the median area. The SEM method provides an array of morphological features for use in identifying fly eggs. However, this method does have some disadvantages. The main disadvantage is that it requires expensive equipment and can take time to identify the species from which the egg originated, so it may not be useful in a field study or to quickly identify a particular egg. 58. The SEM method is effective provided there is ample time and the proper equipment and the particular fly eggs are plentiful. The ability to use these morphological differences gives forensic entomologists a powerful tool that can help with estimating a post-mortem interval, along with other relevant information, such as whether the body has been disturbed post-mortem. Potassium permanganate staining. When scanning electron microscopy is not available, a faster, lower-cost technique is potassium permanganate staining. The collected eggs are rinsed with a normal saline solution and placed in a glass petri dish. The eggs are soaked in a 1% potassium permanganate solution for 1 minute and then dehydrated and mounted onto a slide for observation. 58. These slides can be used with any light microscope with a calibrated eyepiece to compare various morphological features. The most important and useful features for identifying eggs are the size, length, and width of the plastron, as well as the morphology of the plastron in the area around the micropyle. 58. The various measurements and observations when compared to standards for forensically important species are used to determine the species of the egg. Mitochondrial DNA. In 2001, a method was devised by Jeffrey Wells and Felix Sperling to use mitochondrial DNA to differentiate between different species of the subfamily Chrysomeinae. This is particularly useful when working to determine the identity of specimens that do not have distinctive morphological characteristics at certain life stages. 59. Mock crime scenes. A valuable tool that is becoming very common in the training of forensic entomologists is the use of mock crime scenes using pig carcasses. The pig carcass represents a human body and can be used to illustrate various environmental effects on both arthropod succession and the estimate of the post-mortem interval. 60. Pigs are the most frequently utilized model in an attempt to gather data regarding forensic experimental analysis. The latter is highly proportionate to human nature due to our overlapping characteristics with the mentioned species. These interrelated components include subcutaneous fat stores, skin thickness, range of adult body mass, hair covering, and omnivorous diets. 61. Gene expression studies. Although physical characteristics and sizes at various instars have been used to estimate fly age, a more recent study has been conducted to determine the age of an egg based on the expression of particular genes. This is particularly useful in determining developmental stages that are not evidenced by change in size, such as the egg or pupa, and where only a general time interval can be estimated based on the duration of the particular developmental stage. This is done by breaking the stages down into smaller units separated by predictable change in gene expression. 62. 3 genes were measured in an experiment with Drosophila melanogaster, bicoid BCD, slalom SLL, and chitin synthase CS. These 3 genes were used because they are likely to be in varied levels during different times of the egg development process. These genes all share a linear relationship in regards to age of the egg, that is, the older the egg is the more of the particular gene is expressed. 62. However, all of the genes are expressed in varying amounts. Different genes on different loci would need to be selected for another fly species. The gene's expressions are mapped in a control sample to formulate a developmental chart of the gene expression at certain time intervals. This chart can then be compared to the measured values of gene expression to accurately predict the age of an egg to within 2 hours with a high confidence level. 62. Even though this technique can be used to estimate the age of an egg, the feasibility and legal acceptance of this must be considered for it to be a widely utilized forensic technique. 62. One benefit of this would be that it is like other DNA-based techniques, so most labs would be equipped to conduct similar experiments without requiring new capital investment. This style of age determination is in the process of being used to more accurately find the age of the instars and pupa. However, it is much more complicated, as there are more genes being expressed during these stages. 62. The hope is that with this and other similar techniques a more accurate PMI can be obtained. DNA analysis. Modern forensic entomology now relies heavily on DNA analysis as a fundamental tool for accurately identifying insect species and gaining important insights into their interactions with human remains. Using this method, DNA is extracted from insect specimens discovered at crime scenes and compared to databases containing known DNA sequences. Forensic entomologists can verify species identification, identify the existence of particular species linked to decomposition, and even establish a connection between insects and particular geographical areas through the examination of insect DNA. 63. The identification of insect stomach contents is one of the main uses of DNA analysis in forensic entomology. Investigators can ascertain the insect's most recent meal by sequencing the DNA contained in the gut of maggots or other insect larvae discovered on a body. When determining the postmortem interval (PMI) or locating possible sources of contamination or infection, this information might be extremely important. 40. The study of insect dispersal patterns and colonization behavior has also been transformed by DNA research. Researchers can deduce patterns of travel and colonization by examining the genetic variety of insect populations. This can provide important insights into the origin of insect specimens found at crime scenes or the transportation of a body. 59. In general, DNA analysis has improved forensic entomology's accuracy and dependability significantly, allowing investigators to obtain previously unobtainable specific information from insect evidence. Stable isotope analysis. A contemporary method that is being used more and more in forensic entomology to shed light on the ecology and life cycle of insects connected to human remains is stable isotope analysis. Using this technique, the stable isotopic composition of elements found in insect tissues, including carbon, nitrogen, hydrogen, and oxygen, is measured. The environment in which the insect developed, including its diet and place of origin, is reflected in these isotopes. 64. Stable isotope analysis provides information on the travel of the body or the insect itself, and can be used in forensic investigations to assist identify the geographic origin of insects found on a body. Furthermore, features of the insect's diet, such as whether it consumed organic matter or human remains, can be inferred by stable isotope analysis. This information can be vital to comprehending the insect's function in the decomposition process. 65. When using traditional methods to identify insect species or estimate age, this methodology has proven quite helpful. Stable isotope analysis contributes to more accurate and thorough forensic studies by improving the precision and depth of forensic entomological investigations by offering a distinct chemical signature that represents the insect's surroundings. Insect activity case study. A preliminary investigation of insect colonization and succession on remains in New Zealand revealed the following results on decay and insect colonization. 66. Open field habitat. This environment had a daily average maximum temperature of 19.4 degrees Celsius, 66.9 degrees Fahrenheit, and a daily minimum temperature of 11.1 degrees Celsius, 52.0 degrees Fahrenheit. The average rainfall for the first 3 weeks in this environment was 3.0 millimeters per day. Around days 17 to 45, the body began to start active decay. During this stage, the insect succession started with Calliphora stygia, which lasted until day 27. The larvae of Chrysomia rufifacies were present between the day 13 and day 47. Hydrotia rostrata, larvae of Lucilia sericata, family Psychotidae, and Silvicola were found to be present relatively late in the body's decay. Coastal sand dune habitat. This environment had an average daily maximum temperature of 21.4 degrees Celsius, 70.5 degrees Fahrenheit, and minimum of 13.5 degrees Celsius, 56.3 degrees Fahrenheit. The daily average rainfall was recorded as 1.4 millimeters per day for the first 3 weeks. The post-decay time interval, beginning at day 6 after death and ending around day 15 after death, is greatly reduced from the average post-decay time, due to the high average temperature of this environment. Insects obtained late in the post-active stage include the Calliphora quadrimaculata, adult Sphaeroceridae, Psychotidae and Piophilidae. No larvae from this last family were obtained in recovery. Native bush habitat. This environment had recorded daily average maximum and minimum temperatures were 18.0 and 13.0 degrees Celsius, 64.4 and 55.4 degrees Fahrenheit respectively. The average rainfall in this habitat was recorded at 0.4 millimeters per day. After the bloat stage, which lasted until day 7 after death, post-active decay began around day 14. In this habitat, the H. rostrata, adult Phoridae, Silvicola larvae and adult were the predominant species remaining on the body during the pre-skeletonization stages. In literature. Over time, the study of forensic entomology evolved from an esoteric science reserved only for entomologists and forensic scientists. Early 20th century popular scientific literature began to pique a broader interest in entomology. The very popular 10-volume book series, Alfred Graham's Thirlbin, Life of Animals, 1876-1879, expounded on many zoological topics, including the biology of arthropods. The accessible writing style of French entomologist Jean-Henri Fabre was also instrumental to the popularization of entomology. His collection of writings Souvenirs entomologiques, written during the last half of the 19th century, is especially useful because of the meticulous attention to detail to the observed insects' behaviors and life cycles. 667. The real impetus behind the modern cultural fascination with solving crime using entomological evidence can be traced back to the works Faune des Tombeaux, Fauna of the Tombs, 1887, and Les Faunes des Cadavres, Fauna of Corpses, 1894, by French veterinarian and entomologist Jean-Pierre Magnin. These works made the concept of the process of insect ecological succession on a corpse understandable and interesting to an ordinary reader in a way that no other previous scientific work had done. It was after the publication of Magnin's work that the studies of forensic science and entomology became an established part of Western popular culture, which in turn inspired other Western scientists to continue and expand upon his research. 6.5. See also. Forensic entomological decomposition. Forensic entomological decomposition is how insects decompose and what that means for timing and information in criminal investigations. Medico-legal entomology is a branch of forensic entomology that applies the study of insects to criminal investigations, and is commonly used in death investigations for estimating the post-mortem interval PMI. 1 method of obtaining this estimate uses the time and pattern of arthropod colonization. 3. This method will provide an estimation of the period of insect activity, which may or may not correlate exactly with the time of death. 1. While insect successional data may not provide as accurate an estimate during the early stages of decomposition as developmental data, it is applicable for later decompositional stages and can be accurate for periods up to a few years. 4. Decomposition. Decomposition is a continuous process that is commonly divided into stages for convenience of discussion. 5. 6. When studying decomposition from an entomological point of view and for the purpose of applying data to human death investigations, the domestic pig Sus scrofa Linnaeus is considered to be the preferred human analogues. 2. 5. 6. When studying decomposition from an entomological point of view and for the purpose of applying data to human death investigations, the domestic pig Sus scrofa Linnaeus is considered to be the preferred human analogues. 2. In entomological studies, 5 stages of decomposition are commonly described. 1. Fresh, 2. Bloat, 3. Active decay, 4. Advanced or post-decay, and 5. Dry remains. 2. 7. While the pattern of arthropod colonization follows a reasonably predictable sequence, the limits of each stage of decomposition will not necessarily coincide with a major change in the faunal community. Therefore, the stages of decomposition are defined by the observable physical changes to the state of the carcass. 8. A pattern of insect succession results as different carrion insects are attracted to the varying biological, chemical and physical changes a carcass undergoes throughout the process of decay. 2. A decaying carcass provides a temporarily, rapidly changing resource which supports a large, dynamic arthropod community. M. Grasberger and C. Frank. Fresh stage. Pig carcass in the fresh stage of decomposition. The fresh stage of decomposition is generally described as the period between the moment of death and when the first signs of bloat are apparent. 2. 6. There are no outward signs of physical change, though internal bacteria have begun to digest organ tissues. 4. No odor is associated with the carcass. 2. 6. Early post-mortem changes, used by pathologists as medical markers for early post-mortem interval estimations, have been described by Goff and include liver mortis, rigor mortis and alger mortis. The first insects to arrive at decomposing remains are usually Caliphoridae, commonly referred to as blowflies. These flies have been reported to arrive within minutes of death or exposure, and deposit eggs within 1 to 3 hours. Adult flies of the families Sarcophagidae, flesh flies, and Mussidae are also common in this first stage of decomposition. First eggs are laid in or near the natural orifices of the head and anus, as well as at the site of perimortem wounds. 2. Depending on the rate of decomposition and the development time of particular blowfly species, eggs may hatch and young larvae begin to feed on tissues and liquids while the carcass is still classified in the fresh stage. 9. Adult ants may also be seen at a carcass during the fresh stage. Ants will feed both on the carcass flesh as well as eggs and young larvae of first arriving flies. 5. Bloat stage. Pig carcass in the bloat stage of decomposition. The first visible sign of the bloat stage is a slight inflation of the abdomen and some blood bubbles at the nose. 5. Activity of anaerobic bacteria in the abdomen create gases, which accumulate and results in abdominal bloating. 2. A color change is observed in the carcass flesh, along with the appearance of marbling. During the bloat stage the odor of putrefaction becomes noticeable. 6. Blowflies remain present in great numbers during the bloat stage, and blowflies, flesh flies and mussids continue to lay eggs. Insects of the families Piophilidae and Phanidae arrive during the bloat stage. Ants continue to feed on the eggs and young larvae of flies. 5. 6. The first species of Coleoptera arrive during the bloat stage of decomposition, including members of the families Staphylinidae, robe beetles, Sylphidae, carrion beetles, and Claridae. These beetles are observed feeding on fly eggs and larvae. 2. 6 beetle species from the families Hysteridae may also be collected during this stage, and are often hidden beneath remains. 5. 6. Active decay stage. Pig carcass in the active decay stage of decomposition. The beginning of active decay stage is marked by the deflation of the carcass as feeding dipteran larvae pierce the skin and internal gases are released. During this stage the carcass has a characteristic wet appearance due to the liquefaction of tissues. Flesh from the head and around the anus and umbilical cord is removed by larval feeding activity. 5. A strong odor of putrefaction is 5. A strong odor of putrefaction is associated with the carcass. 2. Feeding larvae of Caliphoridae flies are the dominant insect group at carcasses during the active decay stage. 2. At the beginning of the stage larvae are concentrated in natural orifices, which offer the least resistance to feeding. Towards later stages, when flesh has been removed from the head and orifices, larvae become more concentrated in the thoracic and abdominal cavities. 5. Adult Caliphorids and mussids decreased in numbers during this stage, and were not observed to be mating. 5. However, non-Caliphoridae dipterans are collected from carcasses. 2. The first members of Sepsidae arrive at the carcass during the active decay stage. Members of Coleoptera become the dominant adult insects at the site of remains. In particular, the numbers of Staphylinids and Hysterids increase. 5. Advanced decay stage. Pig carcass in the advanced decay stage of decomposition. Pig carcass in the dry remains stage of decomposition. Blowfly and fly larvae on 5-day-old corpse of South African porcupine, Hystrix africaestralis. Most of the flesh is removed from the carcass during the advanced decay stage, though some flesh may remain in the abdominal cavity. Strong odors of decomposition begin to fade. 2. 5. This stage marks the first mass migration of third instar Caliphorid larvae from the carcass. Piophilidae larvae may also be collected at this stage. 2. 6. Few adult Caliphoridae are attracted to carcasses in advanced decay. Adult Dermestidae, skin beetles, arrive at the carcass. 6. Adult dermestid beetles may be common, whereas larval stages are not. 2. Dry decay. The final stage of decomposition is dry remains. Payne described a total of 6 stages of decay, the last 2 being separate dry and remains. As these stages are nearly impossible to distinguish between, many entomological studies combine the two into a single final stage. Very little remains of the carcass in this stage, mainly bones, cartilage and small bits of dried skin. There is little to no odor associated with remains. 2. 6. Any odor present may range from that of dried skin to wet fur. 5. The greatest number of species are reported to occur in the late decay and dry stages. 2. 5. The dry decay stage is characterized by the movement from previously dominant carrion fauna to new species. 5. Very few adult caliphorids are attracted to the carcass at this stage. 6. And adult piophilids emerge. 2. The dermestid beetles, common in advanced decay, leave the carcass. Non-carrion organisms that commonly arrive at remains in dry decay are centipedes, millipedes, isopods, snails and cockroaches. 5. Factors affecting decomposition. Understanding how a corpse decomposes and the factors that may alter the rate of decay is extremely important for evidence in death investigations. Campo Basso, Vela, and Introna consider the factors that may inhibit or favor the colonization of insects to be vitally important when determining the time of insect colonization. 10. Temperature and climate. Low temperatures generally slow down the activity of blowflies and their colonization of a body. Higher temperatures in the summer favor large maggot masses on the carrion. Dry and windy environments can dehydrate a corpse, leading to mummification. Dryness causes cessation in bacterial growth since there are no nutrients present to feed on. Access. Access to the body can limit which insects can get to the body in order to feed and lay eggs. Those circumstances that enhance the availability of corpses for arthropod colonization are called physical barriers. For example, corpses found in brightly lit areas are generally inhabited by Lucilia illustris, in contrast to Phormia regina, which prefers more shaded areas. Darkness, cold, and rain limit the amount of insects that would otherwise colonize the body. A submerged corpse can vary in temperature and is colonized by very few terrestrial insects. Fish, crustaceans, aquatic insects 11, 12, and bacteria would be the likely fauna in this case. Bodies that have been buried are harder to get to than freely available bodies, which limits the availability of certain insects to colonize. The coffin fly Megacelia scalaris is one of the few fly species seen on buried bodies because it has the ability to dig up to 6 feet underground to reach a body and oviposit. Reduction and cause of death. Scavengers and carnivores such as wolves, dogs, cats, beetles, and other insects feeding on the remains of a carcass can make determining the time of insect colonization much harder. This is because the decomposition process has been interrupted by factors that may speed up decomposition. Corpses with open wounds, whether pre or post-mortem, tend to decompose faster due to easier insect access. The cause of death likewise can leave openings in the body that allow insects and bacteria access to the inside body cavities in earlier stages of decay. Flies oviposit eggs inside natural openings and wounds that may become exaggerated when the eggs hatch and the larvae begin feeding. Clothing and pesticides. Wraps, garments, and clothing have shown to affect the rate of decomposition because the corpse is covered by some type of barrier. Wraps, such as tight fighting tarps, can advance the stages of decay during warm weather when the body is outside. However, loose-fitting coverings that are open on the ends may aid colonization of certain insect species and keep the insects protected from the outside environment. This boost in colonization can lead to faster decomposition. Clothing also provides a protective barrier between the body and insects that can delay stages of decomposition. For instance, if a corpse is wearing a heavy jacket, this can slow down decomposition in that particular area and insects will colonize elsewhere. Bodies that are covered in pesticides or in an area surrounded in pesticides may be slow to have insect colonization. The absence of insects feeding on the body would slow down the rate of decomposition. Percent body fat of corpse. More fat on the body allows for faster decomposition. This is due to the composition of fat, which is high in water content. Larger corpses with higher percent body fat also tend to retain heat much longer than corpses with less body fat. Higher temperatures favor the reproduction of bacteria inside high nutrient areas of the liver and other organs. Drugs. On occasion, drugs that are present in the body at death can also affect how fast insects break down the corpse. Development of these insects can be sped up by cocaine and slowed down by drugs containing arsenic. 10.13. Current research. New research in the related field entomotoxicology is currently studying the effects of drugs on the development of insects who have fed on the decomposing tissue of a drug user. The effects of drugs and toxins on insect development are proving to be an important factor when determining the insect colonization time. It has been shown that cocaine use can accelerate the development of maggots. In one case, Lucilia serracata larva that fed in the nasal cavity of a cocaine abuser grew over 8 mm longer than larvae of the same generation found elsewhere on the body. 14 other researchers in entomotoxicology are developing techniques to detect and measure drug levels in older fly pupae. This research is useful for determining cause of death for bodies that are found during later stages of decay. To this date, bromazepam, levomepromazine, malathion, phenobarbital, trizolam, oxazepam, alamamazine, clomipramine, morphine, mercury, and copper have been recovered from maggots. 15. Conclusion. Understanding the stages of decomposition, the colonization of insects, and factors that may affect decomposition and colonization are key in determining forensically important information about the body. Different insects colonize the body throughout the stage of decomposition. 2. In entomological studies, these stages are commonly described as fresh, bloat, active decay, advanced decay, and dry decay. 2. 5. Studies have shown that each stage is characterized by particular insect species, the succession of which depends on chemical and physical properties of remains, rate of decomposition, and environmental factors. 5. Insects associated with decomposing remains may be useful in determining post-mortem interval, manner of death, and the association of suspects. 15. Insect species and their times of colonization will vary according to the geographic region. 2. And therefore may help determine if remains have been moved. 15. Reference. Entomotoxicology. In forensic entomology, entomotoxicology is the analysis of toxins in arthropods, mainly flies and beetles, that feed on carrion. Using arthropods in a corpse or at a crime scene, investigators can determine whether toxins were present in a body at the time of death. This technique is a major advance in forensics. Previously, such determinations were impossible in the case of severely decomposed bodies devoid of intoxicated tissue and bodily fluids. Ongoing research into the effects of toxins on arthropod development has also allowed better estimations of postmortem intervals. Effects of toxins on arthropods. Drugs can have a variety of effects on development rates of arthropods. Morphine, heroin, cocaine, and methamphetamine are commonly involved in cases where forensic entomology is used. The stages of growth for insects provides a basis for determining a cause in altered cycles in a specific species. An altered stage in development can often indicate toxins in the carrion on which the insects are feeding. Beetles, order, Coleoptera, and beetle feces are often used in entomotoxicology, but the presence of toxins is often the result of the beetles feeding on fly larvae that have been feeding on the carrion containing toxic substances. Flies, order, Diptera, are the most commonly used insect in entomotoxicology. 1. Through the study of Sarcophaga Caranea tibialis larvae, barbiturates were found to increase the length of the larval stage of the fly, which will ultimately cause an increase in the time it takes to reach the stage of pupation. 2. Morphine and heroin were both believed to slow down the rate of fly development. 3. 4. However, closer examination of the effects of heroin on fly development has shown that it actually speeds up larval growth and then decreases the development rate of the pupal stage. This actually increases the overall timing of development from egg to adult. Research of Lucilia serracata Diptera, Calliphoridae, reared on various concentrations of morphine-injected meat, found higher concentrations of morphine in shed pupal casings than in adults. 5. Cocaine and methamphetamine also accelerate the rate of fly development. 3. Some effects depend on the concentration of the toxin while others simply depend on its presence. For example, cocaine at the lethal dose causes larvae to develop more rapidly 36 to 76 hours after hatching. 3. The amount of growth depends on the concentration of cocaine in the area being fed upon. The amount of methamphetamine, on the other hand, affects the rate of pupal development. A lethal dose of methamphetamine increases larval development through approximately the first 2 days, and afterwards the rate drops if exposure remains at the median lethal dosage. The presence of methamphetamine was also found to cause a decrease in the maximum length of the larva. 3. Along with changes in development rates, extended periods of insect feeding refrain and variation in the size of the insect during any stage of development can also indicate the presence of toxic substances in the insect's food source. 3. Examples of use. Since J.C. Bayer and his partners first demonstrated the ability of toxins to be recovered from maggots feeding on human remains in 1980, the use of entomotoxicology in investigations has made an emergence into the field of forensic entomology. 6. An example of one such case involved the discovery of a 22-year-old female with a history of suicide attempts found 14 days after her death. Due to the body's advanced stage of decomposition, no organ or tissue samples were viable to screen for toxins. Through gas chromatography (GC) and thin layer chromatography (TLC) analysis of Cochleomia micellaria Diptera, Calliphoridae larva found feeding on the woman's body, phenobarbital was detected and perceived to have been in the woman's system upon death. 3. Drug abuse detected. In France, Pascal Kintz and his colleagues were able to demonstrate the use of entomotoxicology to detect toxins that were not discovered during the analysis of body tissues and fluids of a body found roughly 2 months after death. A liquid chromatography analysis on organ tissue and Calliphoridae larva found at the scene revealed the existence of 5 prescription medications. Triazolam, however, was only detected in the analysis of maggots and not in organ tissue samples. Comparative research showed increased sensitivity of toxicological analysis of Diptera samples over decomposed body tissues. A similar case involved the discovery of the remains of a 29-year-old known to abuse drugs, last seen alive 5 months prior. Through the use of GC and GCMS techniques, Nolt and his partners discovered the presence of cocaine in decomposed muscle tissue and in maggots found on the body. However, due to the severity of decomposition of the muscle tissue, more suitable drug samples devoid of decomposition byproducts were reared from the maggots. 3. Aid determination of origin. Pekka Nuortiva presented the case of a young woman found severely decomposed in Inga, Finland. Diptera larvae recovered from the body were reared to adulthood and found to contain low levels of mercury, indicating that the woman came from an area of comparatively low mercury pollution. This assumption was proven correct once the woman was identified and found to have been a student in Turku, Finland. This case demonstrated the ability of toxicological analysis to help determine origin. 7. This case applied Nuortiva's research involving mercury and its effect on maggots. Through experimentation, Pekka Nuortiva presented the case of a young woman found severely decomposed in Inga, Finland. Diptera larvae recovered from the body were reared to adulthood and found to contain low levels of mercury, indicating that the woman came from an area of comparatively low mercury pollution. This assumption was proven correct once the woman was identified and found to have been a student in Turku, Finland. This case demonstrated the ability of toxicological analysis to help determine origin. 7. This case applied Nuortiva's research involving mercury and its effect on maggots. Through experimentation, it was determined that maggots fed on fish containing mercury possessed levels of mercury in their tissue of even greater concentration than in the tissue of the fish. Nuorteva also discovered that the presence of mercury in the maggots' systems hindered their ability to enter into the pupal stage. 8. Toxin confounding of postmortem interval estimate. Through the analysis of specific cases, it was revealed that toxins present in a person's body upon death can confound postmortem interval estimations. An example of such a case, reported by Gunadalake and Goff, concerned the discovery of a 58-year-old male with a history of attempted suicides found dead in a crawl space in Honolulu, Hawaii, last seen 8 days prior. 2 species of Diptera califorida, Chrysomia megacephala and Chrysomia rufifacies, found on the corpse and tissue samples from the body revealed malathion. Investigators found it abnormal that, given the conditions, there were only 2 fly species found on the body and that these species revealed a postmortem interval of 5 days. Thus it was determined that the presence of the organophosphate malathion in the man's system delayed oviposition for a few days. 7. Paul Katz analyzed a case in Spokane, Washington where maggots rendered differing postmortem estimations. A 20-year-old female victim was found stabbed to death and laying in an open environment surrounded by trees. Most of the oldest maggots found on the body were approximately 6 to 7 m long, which suggested that they were roughly 7 days old. There was, however, a very strange exception, which was the retrieval of a 17.7 mm maggot which suggested an age of 3 weeks. After ruling out the possibility that the maggot had traveled onto the corpse from carrion nearby, it was assumed that there was no conceivable way a 3-week-old maggot could have been present on the corpse. Later investigations revealed that the woman had snorted cocaine shortly before her death and that the 17.7 mm maggot must have fed in the woman's nasal cavity. Research revealed that maggot development can be sped up by the ingestion of cocaine. 9. Use of shed casings and insect feces. Not only are tissues from maggots used to detect toxins, shed casings and insect feces have also been used to detect and identify toxins present in corpses upon death. An instance of this finding was demonstrated by Edward McDonough, a medical examiner in Connecticut. A mummified corpse of a middle-aged woman was found inside of her home. Prescription medicine bottles were found with labels identifying the following drugs: ampicillin, Ciclor, doxycycline, erythromycin, Elavil, Lomadil, pentazocine, and Tylenol 3. McDonough performed toxicological analyses on stomach contents and dried sections of brain and found lethal levels of amitriptyline and nortriptyline. Insect feces, shed pupal cases of Megacelia scalaris, Diptera, Phoridae, and shed larval skins of Dermestes maculates, Coleoptera, Dermestidae, were gathered from the corpse at the scene. McDonough sent these to an FBI lab which broke down the complex structures of the samples using strong acids and bases and freed the toxins for analysis. The cast pupal cases and larval skins were also found to contain amitriptyline and nortriptyline. Larger concentrations were discovered in the pupal cases because phorid flies prefer to feed on softer tissues. The hide beetle larval skins revealed lower concentrations of the drugs because these beetles prefer to feed on dry, mummified bodies. The use of pupal cases and larval skins allows investigators to detect toxins in a body years after death. 8. Limitations. Further research should be conducted in order to fill the gaps in entomotoxicology. Such areas as bioaccumulation, insect metabolism of drugs, and quantitative analyses of insect evidence have only begun to be researched. Because it is a relatively new branch of forensic entomology, entomotoxicology has its limitations. According to Pounder's research, there is no correlation between the drug concentration in tissue and the larva feeding on that tissue. 6. Entomological specimens make for excellent qualitative toxicological specimens. There is, however, a lack of research in the way of developing an assessment that can quantify the concentration of a drug in tissue using entomological evidence. One reason for this is that a drug can only be detected in larvae when the rate of absorption exceeds the rate of elimination. 3. Demonstrated this theory using Calliphora vicina larvae reared on human skeletal muscle obtained from cases of coproximal and amitriptyline overdose. Samples of pupae and third instar larvae no longer contained concentrations of the drugs, suggesting that drugs do not bioaccumulate over the entire life cycle of larvae. This leads entomologists to theorize that toxins are eliminated from the larva's system over time if they are not receiving a constant supply of the toxin. Forensic entomology and the law. Forensic entomology deals with the collection of arthropodic evidence and its application, and through a series of tests and previously set rules, the general admissibility of said evidence is determined. Forensic entomology may come into play in a variety of legal cases, including crime scene investigation, abuse and neglect cases, accidents, insect infestation, and food contamination. Scientific evidence. The admissibility of forensic evidence is left up to the judgment of the court. To provide a strong basis of admissibility for the evidence, accurate documentation is essential so that there is no room for speculation as to the authenticity of the evidence. Given that admissibility is granted, expert witnesses may be called to a courtroom to either support or refute the conclusions that are derived from the evidence submitted. In order to determine what is and is not scientific evidence, a set of rules or parameters must be established for qualification. There are 2 basic principles that govern the acceptability of evidence. Frye test. One of these qualifying tests is the Frye, General Acceptability, test. The Frye standard is the test encountered more commonly in most state proceedings. The Frye standard states that the scientific community must only accept the evidence after it is supported by a certain amount of valid information. 1. This test is used frequently to disqualify sketchy information such as bite mark comparisons, due to the scientific community's general belief that this can be a subjective and unreliable source and could lead to false conviction or acquittal if used in court proceedings. 1. Daubert test. The second test is the Daubert relevancy and reliability requirement. This federal test is applied to testimony given by expert witnesses. Despite the Daubert standard's reliability, it is used to a lesser extent in state courts. The information provided by the witness must pass a two-part examination, it must be deemed relevant to the case at hand, and the witness must prove that it came from a reliable source. 1. Evidence presented in court proceedings is subject to extreme scrutiny, so it is very important that the information presented to the judge and jury has been obtained using the scientific method. Although these two decisions have been used as standards for many years, much of the decision about what evidence makes it into the courtroom depends upon the decisions of the presiding judge. Admissibility of evidence and judicial prudence. Information is usually admitted as evidence if it helps to clarify at least one of the 5 components of analyzing physical evidence. The reconstruction of events or places. Identification is used in scientific tests. Recognition by separating relevant and irrelevant information. Individualization by proving it to be unique to a suspect or a victim. Or usage of comparison analysis methods, DNA, fingerprints, etc. 1. All of these categories, though important, have a lot of gray areas, allowing for the judge to have the final say on what a jury can see and hear in regard to a case. 1. Aversion to scientific evidence. Judges tend to use more caution when admitting scientific data than when considering other types of physical evidence. This has been linked to a concern that scientific data is given priority over other forms of information. 1. Jurors have been known to take scientific evidence as fact, so judges must decide how reliable the information is before allowing a jury to take that information into account. Allowing too much scientific data can make a case less subjective and give jurors less of a choice in their deliberation. Therefore, some judges try to limit charts, graphs, and other forms of overwhelming scientific evidence in relation to a crime. This is one of the main reasons that voiceprints and polygraphs have been denied admissibility in the courtroom. 1. They are both subjective components of scientific evidence, but if presented as fact by prosecutors, the jurors may become misinformed on how reliable the data is, and therefore most judges exclude it. These techniques are still used heavily in detective work, even though they are not admissible in court. These techniques help officers to find other types of evidence that can be brought to court and can help to ensure law enforcement that they are following the correct suspect. Other approaches to scientific evidence that may allow its admittance. One way that some judges have attempted to admit scientific evidence is in its raw form. By allowing the jury to draw their own conclusions, without confusing jargon and charts, some judges are more amicable to this approach. 1. This requires a basic level of knowledge on how to read the data, which can be supplemented with an expert witness who may offer their own opinion. This is a less harsh approach to admitting scientific evidence. Judges also place a large weight on the amount of corroborating evidence that can be submitted to support each piece. 1. Typically, a judge will not admit a piece of evidence unless it can be supported by expert testimony, witnesses, or other physical evidence. One piece of evidence in a crime is somewhat meaningless. Many pieces must fit together and support the prosecution's or the defense's theory. Similarly, a judge will not grant admissibility to evidence that proves guilt or innocence without a doubt. 1. This rule is eagerly applied to matters of scientific evidence because juries tend to place too much emphasis on certain evidence, particularly DNA evidence, which is believed to be foolproof. It is preferred that the circumstances are reconstructed and confirmed in small pieces. The admission of these pieces is viewed as unfair to the other counsel and is denied. 1. The goal of the court is to leave a decision to be made by the jury after all of the evidence has been presented. Large, sweeping evidentiary discoveries take the subjectivity of the evidence out, especially in regard to scientific discoveries. Forensic entomology as evidence in court. Using forensic entomology as evidence in criminal proceedings requires the examination of a few other judicial requirements. Federal Rules of Evidence 702 and 703. Most entomological evidence, or evidence dealing with the presence of insect larvae or adults being found at a scene, enters through expert witness testimony. Because there is little common knowledge associated with the discovery of particular larvae at a crime, it is useful for an entomologist to come in and explain what they found and what that could mean in regards to the location of death, the time of death, and other various parts of the case. 1. This type of witness testimony enters under the Federal Rules of Evidence 702 and 703. 1. Daubert v. Merrill Dow Pharmaceuticals. Use of repulsive and inflammatory evidence. One rule that applies very specifically to forensic evidence is that the courts ban inflammatory or repulsive information or photographs. 1. This can be especially difficult for entomologists because photographs of insects feeding on dead bodies tend to be controversial, but they can be crucial in making their part of the case clear. Extreme caution must be used when choosing any photographs of entomological evidence to share with a jury. This rule was put into place in order to avoid outraged jurors. Emotional responses to this type of evidence have proven to distract jurors from the issue at hand, possibly leading them to make decisions based on irrelevant data. 1. Other important considerations. Another important factor in creating admissible entomological evidence is creating a clear and consistent chain of custody. 1. This means that any samples taken from a body or the scene of the crime in question must have been labeled correctly and the identity of those persons who had access to it must be recorded. This record should show the location and custody of the samples at all times, from collection to analysis. Typically, an entomologist on the scene will collect their samples and lock them in their scene kit as a way of making sure that no one else has access to them to tamper with the evidence, ensuring that the information gathered in the field is viable in court. If an accurate record of custody is not kept, then the presentation of this evidence could provide a reason for appeal. Case records and reports. As is the case when collecting and handling evidence, documentation of the source, transfer, and state of said entomological evidence is crucial to a criminal investigation. 2. An accurate record facilitates the tracking of individual pieces of evidence even if the item has been modified after the initial collection. Modification can result by means of natural biological processes, like metamorphosis, or the manipulation of the insects by the lab during analysis. 2. Accurate documentation also helps preserve the chain of custody for the evidence collected. It accounts for a detailed record of who has had access to the evidence, and the exact times and locations of when the evidence was moved and for what reason. It basically provides a step-by-step ranging from the time of collection to the court hearings. Forensic entomologist (FE) records relating to a death scene investigation can be divided into 8 categories: initial contact notes, evidence submitted, death scene case study form, autopsy report, local weather records, specimen disposition and identification record, chain of custody record and receipts, and finally, the case study final report. 2. Initial contact notes. Initial contact notes include certain preliminary information and the fee charged. The following are key pieces of preliminary information: the contact officer's name, address, and phone number, the death scene's exact location, the type of habitat in that area, the corpse's phase of decomposition, and a general description of the manner of death. 2. When recording the information gathered, the estimated post-mortem interval (PMI) made by others at the crime scene should be disregarded in order to guarantee an unbiased PMI from the entomologist. Since entomology expertise is usually requested well after the recovery of the corpse, inquiries about the kind and amount of insect activity observed and whether the body had already been moved should also be recorded. Finally, the entomologist and the law enforcement organization should mutually agree on a clear fee that appropriately covers all the services to be provided by the FE. Evidence submitted. Because it is not always possible for the forensic entomologist to be present at a crime scene to collect the appropriate evidence, it is important that clear and concise instructions are left for the handling of live and preserved specimens to those processing the scene. 2. In these cases where the FE is not present at the scene, the transport of live and preserved specimens is necessary. As a rule, if specimens are in transport longer than an hour, they have to be kept in a chilled insulated container with enough coolant to keep them relatively inactive and stop their development. All evidence should be handled with a reasonable amount of security, from videotapes and photographs to actual specimens recovered from the field. In all cases, the evidence should be secured with a seal that ensures absolutely no tampering. Death scene case study form. As the name suggests, this form is an itemized checklist for the different conditions present at the crime scene. 2. The conditions recorded include temperature, habitats, the presence/absence of sunlight, and other details specific to the crime scene. More often than not, this form merely repeats previously recorded observations. This is because the primary purpose of the death scene case study form is to itemize the different findings to ensure that nothing is overlooked. Autopsy report. The autopsy report is the responsibility of the coroner, not the forensic entomologist, and must be requested. The most important things to note are the manner and cause of death, 2. Where the insects were found on the body, and the extent of decomposition of the body. Because the ME is one of the first people to have access to the body, she is usually the one to retrieve specimen samples from different orifices on the body. The specific location of the insects and the extent of their activity should be duly noted on the report. This information helps the entomologist determine an accurate PMI based on the development of the specimens found. Local weather records. Accurate weather reports are elemental in a death scene investigation, especially one where any type of developmental insect is involved. Even within a relatively small area, the weather can vary, so it is important that the source of climate conditions is extremely reliable and accurate. For an ideal estimation is for the general maturation of the live specimens found on the body, a record of the ground and ambient temperatures should be taken for several days after the corpse is removed from the scene. 2. To ensure the utmost accuracy, it is best to collect weather data from a weather station located as close as possible to the actual crime scene. Specimen disposition and identification record. Most of the specimens collected from a corpse at a crime scene are maggots. Insects found in this immature stage of development are incredibly difficult to identify because all maggots have the same appearance when in this stage. It is because of this that most live specimens collected are reared into their adult stage where identification is increasingly easier. 2. It is in the specimen disposition and identification record that all developmental changes are recorded. Meticulous records of the changes that the specimen undergoes are duly noted and explained. In the same manner, when dissecting the immature insect, proper records must also be taken. Following dissection and proper documentation, all the specimen species are placed together in a labeled vial. 2. Chain of custody record and receipts. Evidence is never in one place only. Eventually, it has to be transferred among 2 or more parties, whether it be between entomologists, from the original site of the crime scene to an analytical lab, or between law enforcement departments. Documentation of all these transfers have to be properly annotated on the chain of custody records to ensure that the chain of evidence is never broken. 2. The receipts serve as verification for the whereabouts of the evidence at all times. Case study final report. The final and most important component of a crime scene's documentation is the case study final report. It is a compilation of all the relevant information gathered in the previously mentioned records and reports. On it, there will be such information as the contact person's name, address, and phone number, a proposed scenario for the crime and all relevant details on the crime scene, as well as the last time the deceased was seen alive. 2. As you recall, this is all information found in the initial contact notes. Other relevant information is a list of all evidence collected, including specimens and all identification and rearing procedures performed, a PMI estimate, and a summarized conclusion based on all the findings. Copies of this report are submitted to the contact officer, the forensic entomologist, and as a courtesy, to the medical examiner. If throughout the course of the investigation, any manipulation of the specimens occurred, i.e. dissecting, rearing, then a copy of the specimen disposition and identification form should also be included in the report. 2. It is important that the FE remains completely objective as she documents his findings and compiles all significant evidence and data into this final report. At times, the evidence may allow for several different interpretations, and to remain objective, the FE must explore all these plausible scenarios. Expert witness. When a forensic entomologist is called to the stand, her expertise is only requested so that she may refute or support the case study final report. It is imperative that objectivity predominates and is not lost as the trial goes on. Although it may be used for referencing and corroboration of evidence, the case study final report is seldom admitted into court proceedings. Preparation for court. The case study final report is the main focus so that ideal preparation may take place. 2. This report does, after all, contain all the entomological findings and conclusions relevant to the case. The report should be detailed thoroughly but should still allow room for speculation because forensic entomology is still a relatively new science, and it would be a bit overzealous for the forensic entomologist to announce full certainty on the subject. Said speculation should not exist when it comes to the evidence. Disclosure of this report to the opposing side before the trial is recommended so that the defense and/or prosecution have time to consult another expert witness to look over the original findings. 2. Another thing is that proper credit should be given where credit is due. If any of the conclusions derived by the forensic entomologist were seconded by another professional, that person's name and qualifications should be presented as well. 2. It is the responsibility of the forensic entomologist to adequately instruct on the collection and handling of all specimen evidence. Even though it is upon them to try to ensure efficient collections, they cannot always prevent inept actions. Because of this, the FE can refuse improper analyses and tests performed on the evidence. 2. It is important that the FE is diligent with the procedures and collection, so that when in court, the opposing side can't question the findings. In court. First of all, appearances in court merit professional attire. Upon arrival, the FE will wait his turn to be called to the stand in a waiting room. After being sworn in, the supporting counsel will ask for the phase qualifications. 2. It is vital that all qualifications, awards, published works, fellowships and grants, and all merit are mentioned because this information is what is going to determine whether the forensic entomologist is qualified enough to be an expert witness. As the expert witness is being walked through the evidence, she has to keep in mind that most people have no thorough knowledge of entomology, so the responses to the questions being asked and the explanations about the evidence have to be kept clear and concise and in a simplistic manner that informs and educates, not only the jurors but the judge as well. It is vital that the attorney has a thorough grasp of all the concepts that are being explored so that she knows which questions to ask to carry across his point. In the same manner, the responses to the questions being asked by the opposing side should be kept as precise as possible, so as not to give the opposing attorney any ammunition to discredit the findings in the cross-examination. If any type of photographic evidence is involved, the expert witness has to request permission from the judge or attorney prior to the trial to make sure that the photographs that are to be presented aren't prejudiced and have the ability to sway the jury. 2. Finally, it is important that the expert witness realizes that replying, "I don't know," to a question is perfectly fine. It is preferable to say that you don't have the expertise in that particular area than to claim that you do and make yourself look like a liar. Before leaving the witness stand, proper notification will be given to the expert witness to see if he is dismissed or if he will be recalled later. 2. Case studies. A good example of a case where forensic entomology was successful in determining a postmortem interval is one involving a young prostitute and a 30-year-old member of the army. A family member had informed the police of her disappearance 4 days before her body was found. She had last been seen in the presence of this military man on May 31st. 3. A large number of adult flies and maggots were noticed and gathered inside and near the wounds of the young girl. A number of these maggots were kept and grown to be adult flies, reared, and others were taken in their present state. Pictures were taken and enlarged showing the flies around the body when it was found. Using this evidence along with correlating weather conditions known to be present around that time, an entomologist was able to give a postmortem interval. 3. The evidence showed that the flies had arrived on May 31st, which indicated a time period of 4 days since she had died. This information showed the army man to be the last person to be with the girl. When presented with the evidence, authorities were able to get a confession out of the man. 3. The use of forensic entomology was definitely a key factor in this case. Another case where forensic entomology was used successfully deals with a body being reported to be thrown down a well. When authorities searched for the well in southern Indiana, it was clear they found the correct one. Thousands of flies were flying over material blocking the well. The pungent smell and the fact that the flies could not access the body created this swarm. 3. Given that vast number of wells in the area, the presence of the flies indicated the body's presence, allowing it to be found in a timely manner. 3. The use of entomology is not only important in determining when a person has died, but also simply locating a corpse. Famous cases. A handful of well-publicized criminal cases helped bring forensic entomology into the limelight and popularized it in the modern era. Paul Bernardo. In 1992, the Canadian press went into a frenzy concerning the Ken and Barbie murders. In June 1991, Paul Bernardo and Karla Homolka tortured, raped, and killed 14-year-old Leslie Mahaffey. The brutal assaults of Mahaffey were recorded on tape by the couple and helped convict them at trial. 10 months later, in April 1992, Bernardo and Homolka again raped, tortured, and killed another teenage victim, Kristen French, over a period of several days, all captured on videotape. 4. Once captured and put on trial, forensic entomology began to play an important role. In Paul Bernardo's trial, renowned forensic entomologist Dr. Neil Haskell helped establish a timeline for one of the victim's murders. Haskell utilized his understanding of insect development cycles to narrow a 15-day time interval down to a period of 4 days for the victim's time of death. Being one of Canada's most publicized criminal cases, Dr. Haskell and his testimony brought forth forensic entomology. 5. David Westerfield. In the United States, forensic entomology became well known through the trial of David Westerfield in 2002 for the abduction and murder of 7-year-old Danielle Van Dam. In this San Diego case, the courtroom became a battle over conflicting reports from 4 forensic entomologists: David Faulkner, Neil Haskell, Emily Goff, and Robert D. Hall. A 5th scientist, William Rodriguez, a forensic anthropologist, also testified on this issue. Danielle had vanished from her bedroom the night of February 1 to 2. Her partially decomposed body was found on February 27. Haskell, Hall, and Faulkner argued that forensic entomology established that the body was dumped in mid-February, during which time Westerfield was already under police surveillance, since February 5, while Goff said the victim's body was colonized by larvae earlier than the other 3 entomologists estimated. His dates were February 9 to 14. 6, 7. His dates were earlier as he used new data from Canada, which gives a significantly slower development for that fly species. 8. Richard Merritt, a forensic entomologist and professor at Michigan State University, stated that because each scientist has his own method, this can lead to disagreement, but a lot of the disagreements involve a variation of 1 or 2 days, not over a week and a half as in the Westerfield case. If it's that big a time, someone screwed up. 9. All the entomologists, including the prosecution's expert Goff, gave dates that excluded the defendant by a significant margin, at least 4 days. Yet the jury rejected that evidence and found him guilty. The foreman explained that he felt the field was too subjective to exonerate Westerfield. 10. The prosecutor, Jeff Dusek, was highly critical of the forensic entomology evidence. In fact, he dismissed the field outright. 11. Other important cases. A case that shaped Canadian judicial history and brought forensic entomology into the headlines was the exoneration of Stephen Truscott 48 years after being indicted for murder. 12. Forensic entomology also played a role in the investigation of the 1993 raid on David Koresh and the Branch Davidians. 5. Due to the publicity given to these cases, forensic entomology was able to seep further into popular culture. In 2004, the first use of forensic entomology in Brazil demonstrated its importance and potential if executed properly. 26 corpses were found in an indigenous region of Rondônia, Brazil. The bodies of coal miners who came in contact with a group of natives and were brutally clubbed and speared were found. The bodies were excavated and later transported to be autopsied. Arrowheads, spears, and clothing were collected, but no larvae, pupae, or casings were recovered from the crime scene. Once the cadavers were autopsied, only 57 larvae were present on the bodies, and unfortunately, a maximum post-mortem interval could not be established. In part because of the humidity and high-temperature environment, the bodies decayed quickly. Since only P. fulvinota, a species that colonizes all stages of decay, was found on the bodies, no timeline of succession could be established. Had more thorough work been done on the part of the investigators or the use of DNA in forensic entomology been employed, a more conclusive post-mortem interval could have been established through the casings of stage-specific arthropods. 13. The CSI effect and the law. From the contributions of movies and television series, forensic interest has increased and multiple branches spur into various fields. Entomology, an important area of research for the agriculture and veterinary sciences, also aids in crime scene investigation and forensic chronology. Coined after the explosively popular CBS series CSI, the CSI effect encompasses the overwhelming impact investigative scientific documentary shows have had on society. Some notable American institutions such as Michigan State University, 14, North Carolina State University, 15, and Texas A&M University, 16, have recently dedicated entire degree paths geared towards training skilled and knowledgeable forensic entomologists. West Virginia University's largest major is forensic science with over 400 undergraduates. 17. From 2000 to 2008, there has been a significant increase in the amount of attention paid by the public to forensic entomology as a science. With more attention given to arthropod development, accurate hypotheses can be formed. By following life cycles and tracking seasonal changes, entomologists are able to establish the location and weather conditions of an area, which are both particularly useful to homicide detectives. 18. However, with global climate change and increasing temperatures, the formulas used to calculate post-mortem intervals may become skewed. 19. Forensic entomologists like Gail Anderson, of Simon Fraser University in British Columbia, Canada, can rely on these formulas to provide certainty in their predictions. It is possible for them to estimate the time of death to the exact day even if the person has been dead for a matter of weeks. 20. The lines blur when pseudoscience dramatized for viewers feeds misinformation and a false sense of security. Problems with jurors arise when they feel unsatisfied. The jury, wanting more conclusive evidence, scrutinizes minute details, always expecting some kind of forensic smoking gun. 21. 22. After watching a season of House MD or CSI, the jurors sometimes feel qualified to make expert decisions. Additionally, TV prejudices could leave some jurors wanting a dramatic experience, which can delay progress. 23. 24. Even before the trial, the CSI effect can train criminals how to remove evidence and how to stage plausible scenarios. Conversely, victims can intentionally leave crucial traces behind. In one case, a victim who knew she was going to be transported, allowed insects to bite her in hopes of being found and traced back to her area. Current research and entomological findings. The vast majority of forensic entomological casework consists of establishing a very concrete timeline of the events involved in the crime scene. The new set of data provided by forensic entomological casework undoubtedly provides a solid case for either the defense or the prosecution. But as seen in a new series of experiments, the evidence may not be as firm as previously suspected. The use of DNA in forensic entomology is a growing area of research that will become more prevalent in cases as more research is done. In the first experiment, DNA was extracted from a series of immature blue-bodied blowflies. Blowfly. The age range of the blowflies varied from first instar larva up to the third day pupal stage. 25. Extracting the target or victim's DNA from the gut of the larva and pupae becomes inconclusive as you reach the third pupal stage. 25. Therefore, as most samples collected from the crime scene can properly denote the time scene death, there is a chance that the results may be skewed due to improper specimens. Once the proper specimens are collected, a series of DNA sequencing tests can be initiated. Several methods of DNA sequencing exist to further analyze the gut material of the insect and eventually track down human microsatellite DNA. The sequencing methods currently utilized by forensic labs include STR or short tandem repeat profiling and HVR or the hypervariable regions of DNA. 26. The current DNA analysis on forensic cases and forensic research indicates that STR profiling is generally successful, whereas HVR sequencing proved to be a better choice in DNA testing, as its results had a higher success rate. 26. In new research on DNA analysis, STR typing of human DNA from fly larvae fed on decomposing bodies, STR profiling created a complete profile in 7 of the 13 cases, and 2 cases displayed an incomplete set of alleles whereas 4 of the cases showed incomplete profiling. On the other hand, HVR sequencing was successful in all cases except 1. 26. So there is a slight margin of error when utilizing the STR profiling method of DNA typing in forensic analysis. Use of DNA in forensic entomology. Forensic entomology has 3 subfields: urban, stored product and medico-criminal entomologies. This article focuses on medico-criminal entomology and how DNA is analyzed with various blood-feeding insects. Forensic entomology can be an important aspect for law enforcement. With the magnitude of information that can be gathered, investigators can more accurately determine time of death, location, how long a body has been in a specific area, if it has been moved, and other important factors. Blood meal extraction. To extract a blood meal from the abdomen of an insect to isolate and analyze DNA, the insect must first be killed by placing it in 96% ethanol. The killed insect can be stored at -20 degrees Celsius until analysis. When it is time for analysis, the DNA must then be extracted by dissecting the posterior end of the abdomen and collecting 25 milligrams of tissue. The cut in the abdomen should be made with a razor blade as close to the posterior as possible to avoid the stomach. 1. Using a DNA extraction kit, the DNA is extracted from the tissue. If the DNA is mixed with samples from more than one individual, it is separated using a species-specific primer. Once extracted and isolated, the DNA sample goes through a polymerase chain reaction, PCR, is amplified and identified. PCR works by analyzing species-specific mitochondrial DNA. PCR is currently the most commonly used method of species identification. This results from the fact that it is very sensitive in that it requires only a small amount of biological material, and can also utilize material that is not particularly fresh. The sample can be frozen and stored while still remaining usable for later PCR. DNA requires 1 hour to reach the abdomen of an insect, so DNA can be amplified 1 to 44 hours after an insect feeds. Some research suggests that the source of a blood meal can be determined up to 2 months post-feeding. To amplify DNA, it must first be denatured by exposing it to a 95 degrees Celsius temperature for 1 minute, followed by 30 cycles of 30-second 95 degrees Celsius exposures. Then denatured DNA is mixed with a specific primer. A chromatograph is conducted on 2% agarose gel, stained, and viewed with UV fluorescence. The DNA is identified by looking for genome-specific repetitive elements and by comparing it with known examples. Hematophagous insects of forensic importance. Humans are constantly fed on by hematophagous, blood-feeding insects. The ingested blood can be recovered and used to identify the person from which it was taken. Bite marks and reactions to bites can be used to place a person in an area where those insects are found. Order Diptera. The following among the flies Diptera have been utilized. 2. Mosquitoes, family Culicidae. Due to erratic feeding habits, mosquitoes can provide valuable DNA evidence. 3. Multiplex PCR enables reliable identification of bitten individuals from just one mosquito, 4 even few days after taking a blood meal. 5. 6. The insects would need to be collected as soon as possible due to the insect's high mobility, digestion of consumed blood, degradation of DNA, and repeated feeding, although dead specimens are also potentially valuable source of evidence DNA. 7. Research is centered on the mosquito due its widespread presence and affinity for feeding on humans. Biting midges, family Ceratopagonidae. Tsetse flies, family Glossinidae. Sheepkeds, family Hippobosidae. Stable and horn flies, family Mussidae. Sand flies, family Psychodidae, subfamily Phlebotominae. Snipe flies, family Raginidae. Black flies, family Simuliidae. Horse flies, family Tabanidae. Order Siphonoptera. Listed here are fleas commonly encountered by humans that could potentially be used for DNA identification. Sticktite and Shigo flea, family Hectopsilidae, formerly Tungidae, cat flea, C. Tennocephalides felis. Northern rat flea, Nosopsilus fasciatus, human flea, Pullix irritans. Oriental rat flea, Xenopsila chepus. Order Hemiptera. Bedbug, Cymex lectularius. Cymex lectularius is an obligate parasite of humans. Testing a sample of a residence's bedbug population and screening for bites could reveal possible recent visitors to the structure, as they have been observed to feed approximately once a week in temperate conditions. 8. A recent re-emergence of bedbug population.